happy to talk about anything that you want to talk about. So first of all, a description of the basic assay to determine the specific activity of MDH. A bunch of what if scenarios when you're sitting at your UV vis spec and you get a spectrum and it looks like this, what does it mean type things. Things to be aware of, the timing of making up oxaloacetate solutions. Oxaloacetate can be pretty finicky, and if you aren't using it fresh, that can be an issue. The idea of making a master mix, the idea of having some store-bought MDH that you can use as a control if you need it. The idea that there are literature values of specific activity for wild type, and this is glycosomal MDH. And so I like to kind of keep that in the back of my brain. And so if we are very much higher than that, taking a look at students' numbers to see, kind of where they went wrong. And if we're a lot lower than that, especially for wild type, thinking about what some of the issues might have been. I know a number of you are working with mutants of MDH, as am I, and just some suggestions in terms of how to work with low activity that I've come up with, but you may have more that you can add in as well. Amy's going to talk about processing data. So there's some mathematical manipulations involved getting from the data that you're going to get on your UV vis spec to specific activity. And depending upon how you want to spend your time in class and depending upon what level class you're teaching, you may not necessarily care that students go through all of those steps in excruciating detail. And so populating some of those formulas in an Excel spreadsheet, Amy's going to talk about. Amy is also working with 2 classes of 24 students at the same time, so 48 students doing cures. And so she's going to talk about some tips on how to scale this for larger classes, and the use of a microplate reader for specific activity that may be really useful for some people. And then again, things that have worked for you, and then other questions that you have. So that's how we're gonna get started. So let me bring up this first one and put it into full screen mode here. So the reaction that we're looking at is written up on top. So malate dehydrogenase, or MDH, catalyzes the reversible conversion of L-malate and NAD+ to oxaloacetate and NADH. When we study this in the lab, we're studying the back reaction, and that's primarily because that's what's thermodynamically favorable. So NADH and oxaloacetate are our reagents in this reaction. And to measure the rate of a particular reaction, you need something to measure a change in. And NADH provides a really nice visual way for us to measure a change in this reaction because it has an absorbance at 340 nanometers. So what we're going to do in this reaction is measure how NADH gets used up. So we'll start at some level of absorbance at 340 nanometers, and it'll decrease over time. And that's what we're going to be monitoring. So just in general, specific activity is the rate of an enzyme catalyzed reaction divided by the concentration of the enzyme. In this case, the rate that we're going to measure is the change in absorbance at 340 nanometers per minute, which is allowing us to monitor NADH. And so what this ends up being in our specific activity calculations is basically the concentration of NADH per minute divided by the concentration of MDH. So the rate we're going to end up getting from our kinetic studies, the concentration of MDH you're going to have to determine before you do these assays. And so there's a couple of ways to do that. For most of you, you will be purifying your malate dehydrogenase from a histidine affinity column. And so it will come out in a solution that contains imidazole in it. Directly monitoring the absorbance at 280 nanometers and using Beer's law isn't going to work for you. If you need to, for some reason, dialyze the imidazole out of your sample, because of what you're doing downstream with your MDH, using the absorbance at 280 nanometers is a great way to determine concentration. And that's what we do in my lab because I'm going to do fluorescent studies of my malate dehydrogenase mutants, which means I've got to get rid of the imidazole. If you don't need to get rid of the imidazole, just doing a Bradford assay is probably the way to go. So a colorimetric assay, you're going to set up a standard curve, and based on your absorbance in that standard curve, you can figure out your concentration of MDH. If you haven't done Bradford assays and want some tips on how to do that, we can send you an information sheet, actually, that I think Chris just updated. So that's set and ready to go. So we're going to primarily be spending our time talking about how to get that quantity in yellow there, the change in absorbance at 340 nanometers per minute, which is the rate of the reaction. So it's funny that Jing is on this call, because this basic assay is the one that Jing sent me when I first started doing these assays. And I think this is something that Jing simplified from a large document that Ellis Bell had put together. So I want to give credit to those two folks as well. So in this particular assay, there's a few stock solutions that you're going to need. One is our phosphate buffer, which is 100 millimolar sodium phosphate at pH 8. The other is the stock solution of NADH, which is 1 millimolar NADH in water. The next is the stock solution of oxaloacetate. That's 2 millimolar oxaloacetate in water. And then the last is your MDH. And again, you're going to need to know what the concentration of that is, either from a Bradford assay or from the absorbance at 280 if you've dialyzed the imidazole out. Again, oxaloacetate seems to be pretty finicky. And so if you've got an afternoon lab that starts at 1 o'clock, this is not a solution that you want to make up at 8 o'clock in the morning before your classes start. This is a solution you want to be making up at 12:50 right before you walk into your lab. Keeping it cold's a good thing. If you see that your data is deteriorating over the course of the afternoon, it may be a stock solution that you've got to make up again. So the oxaloacetate is going to be the finicky one in these stock solutions. So basic procedures for the assay is you're going to have a cuvette, and what size the cuvette is completely depends on what you have and what your spectrometer will hold. Basically, you want to fill it up about 3/4 of the way so that you're sending light through the solution rather than through the top of the cuvette. And so if you're not using a 1.5 mil cuvette, you can just scale all these accordingly. But in my 1.5 mil cuvette, I'll put 500 microliters of my phosphate buffer, 295 microliters of water, and then I'm going to put that in my spectrometer, which is set at 340 nanometers, and I'm going to blank it. Once I do that, I'm going to add 100 microliters of my stock NADH solution, and I'm going to mix that well. And then I'm going to take a spectrum of that at 340 nanometers. This is my no enzyme control, and this is going to basically let me know where I'm starting at in terms of my absorbance at 340 nanometers, which is going to decrease once I put enzyme in. And we'll take a look at what that should look like in a minute. Once I've got that control done, I'm going to add 100 microliters of the oxaloacetate solution and mix it. And then I'm going to add 5 microliters of my MDH solution and mix it. And at this point, you have a trade-off. You're trying to get things done quickly so you don't miss the beginning part of the reaction, but you also want to make sure that your solutions are well mixed. If your solutions are not well mixed, your data is going to be all over the place. And so it's going to take students a little bit of time to figure out how to mix these and get these done quickly. I usually just take a 3 mil disposable plastic pipette and suck it up and down a few times with the cuvette still sitting in the cuvette holder in the spectrometer, and immediately when I'm done, hit go. In terms of how much MDH you want to add, it's going to be 5 microliters. A good place to start is to aim to have the final concentration in the cuvette being about 0.005 to 0.01 micrograms per mil. You may need to play with that, and I'll show you what I mean by that in a minute. But once these are in, record the data, usually recording it for about 30 minutes and then take the slope from about 3 to 15 seconds, somewhere where it's linear, and this is the initial rate. And then the bottom there says, if your rate's not linear, adjust the amount of enzyme used for the assay and try again. And what I found is that in working with students, this takes a lot longer than I think it will take. And so if you can have one lab period where they're just playing with this, just getting comfortable with this, just figuring out how to get data and how to get on the curve, so to say, And then the next lab period, they can actually collect 3 or 4 or 5 replicates of that data so that they can have some error analysis. That's kind of a good compromise. Some of the students will make it through in one lab period, some just will not. And instead of, kind of telling them too handedly what to do, I like to let them kind of experiment at this point. Okay, so what should this look like? So, all right, so I'm going to do a number of what if scenarios. So you have your 1.5 mil cuvette, you've added 500 microliters of your phosphate buffer, 295 of DI water, you've zeroed the spec, you added 100 microliters of your NADH, you hit go, and you get the spectrum on the left. So at this point, there is no absorbance at 340 nanometers. At this point, there's a problem. The spec's set wrong, the cuvettes are in the backwards, you don't have NADH. My students one day decided to pick up a bottle of DTT instead of NADH, and so doing this no enzyme control is a really good way to start, just to make sure that you've got all of your systems set and ready to go. And when I do this, my absorbance at 340 is somewhere between like 0.4, 0.5, 0.6. And so that's a starting place for us, and that's where we're going to start, and that's where we're going to see the absorbance drop from once we add enzyme. So what should this look like? It should look like this. Got a straight line, again, 0.4, 0.5, 0.6, somewhere in that range, and it's just a straight line. Okay, so you're ready to look at your enzyme activity at this point. So in addition to that NADH, you now add your oxaloacetate stock solution, you add 5 microliters of your MDH. If you get a spectrum that's on the left, this is I've commonly seen this, and the students' first instincts at this point is that the enzyme is dead. There's nothing in there, the line is flat, it's at 0. Well, but you started with the line at like 0.4, 0.5, 0.6. So you've gone from 0.4, 0.5, 0.6 down to 0. So that tells you that you do actually have some activity. The NADH is disappearing. It's just disappearing at a rate that's so fast that it's too fast for you to collect it in the time of your spectrometer. And so in this case, this is telling you you've got way too much MDH in the solution. Go back and do a dilution of your MDH. And this is kind of a repetitive process until students can get on the curve. Okay, so what if you did your NADH control, you added oxaloacetate, you added your MDH, and you get a line that looks like this, where that absorbance that you're seeing is the exact same absorbance as what you had of NADH. This is the case where your enzyme isn't looking like it's in such good shape. There's no perceptible rate that you're measuring there. It may be that you diluted your enzyme too much. So if you're not using full strength enzyme, go back and use a more concentrated version. It may also be, if you're working with a mutant, that you just have something that's really not very active. And so at this point, really what you can do is, if your enzyme is as concentrated as it can be, extend the time and see if you can measure any rate. And we'll talk about that a little bit at the end when we talk about what to do with some of the lower activity mutants. Once one of your students gets a curve that looks like this, they're getting closer. So they've added their NADH, they've added their oxaloacetate, they've added their enzyme, and you can see a decrease in the absorbance at 340 nanometers. But again, before you get to 15 seconds, it's linear down at the baseline. And so in this point, they're close. Their enzyme is still just a little bit too concentrated. The rate is still a little bit too fast for what you are trying to measure. So having them, again, back off on the concentration of enzyme that they have in their cuvette by diluting it a little bit more. And what they should get eventually is something that looks like this. So you've got a slope that's decreasing because you're watching the absorbance at 340 nanometers decrease as you use up oxaloacetate. It's linear over about 15 seconds, and you can measure the slope of that, and that's your rate. So this is what students are looking for. And again, it's going to take a little bit of time for them to figure out how to get on the curve. but that's what that looks like. And so things to be aware of, things to talk about. So again, the oxaloacetate solutions we've talked about, just making sure that they're fresh and that they're cold. I like to have some store-bought MDH around just in case the enzyme that I'm using is dead. It helps me problem solve. And I said, put the store-bought MDH in the freezer. And so I want to talk about that. So the MDH that you've purified from your histidine column, you don't want to freeze. And if you do freeze it, we have, you can put it in solutions with glycerol and certain cryoprotectants. And we have protocols that we can share with you on how to do that. But you're not just going to want to take what comes off your hist column and put it in the freezer. The MDH, the store-bought MDH that I have, I got from Sigma. It's porcine heart. And it's way too much for me to use ever. And so what I do is, you know, it comes as a lyophilized powder. I add water to it and I use what I need. I leave some in the refrigerator for what I'm going to use immediately. And then the rest I put in the freezer, and I'm still taking it out of the freezer 2 and 3 years later, and it's still okay. So I don't know if it has full activity at this point because I don't care. I'm just using it as a control for some activity if my enzyme assay is not working. But I just wanted to say that that caveat about how to store your enzyme. Again, the wild type watermelon MDH specific activity value of 65,000 to 80,000 per minute can help you figure out if your students are in the right range or not. Some of you have asked about whether or not it's okay to make a master mix. The fewer times that students have to pipette, the fewer pipetting errors are into the data that we get, the cleaner the data should be. And so it's definitely possible to add the phosphate buffer, the things that are in purple here, the phosphate buffer, the water, and the NADH are fine in a stock solution that you make up on the day of your lab. Somebody yesterday also said that they added their oxaloacetate into that solution, and it's okay. There's a little bit of a reaction that happens there. And as long as you measure the rate of that little bit of reaction that happens in that master mix and subtract it out from your enzyme reaction, you should be fine. Or you may decide it's so small that you don't even care. I like to keep the oxaloacetate out just because it's pretty finicky. But those are some ways that you can go. And then there were questions on how to work with mutants with low activity. And so if I look at what's in the cuvette that I've used, the 5 things that are colored there are in there. And so if I work to keep the concentration of the buffer constant, I figure that I'm going to add 5 microliters of MDH and I'm going to add 295 microliters of water. So there's no reason why, if I have an enzyme, a mutant that's not really active, I couldn't just add 300 microliters of the mutant and still not change the concentration of NADH or OAA or the concentration of phosphate in the reaction mix. And so that's what I usually do. So when I'm working with mutants, I usually work with a wild type and a mutant at the same time. Whatever I've just done for my wild type, I'll do for my mutant as well. And most of the time, my mutants, it's just going to be slower than my wild type is. And so I've got to figure out how to get more MDH in the cuvette. So instead of diluting it, I'll use it at full strength. And if 5 microliters full strength still isn't great, I'll go ahead and use 300 microliters of the MDH, of the mutant MDH. And at this point, there's no mutant that I've made that has been stable enough to purify that I haven't been able to get kinetics data with using 300 microliters of the mutant. There's some mutants that I've made that have been so unstable that I can't purify them and they express lousy, and I mean, I'm not going to get any data on them at all. But for the ones that are just not real active, this seems to work okay. And so it's a place to start if you're working with something that's not especially active. Okay.
Great. So you had a couple of questions in the chat, and one of them was from Delphine about mutants. Delphine, did you have more questions, or did she answer that question?
Okay. Other questions?
I'm good, thanks.
Okay. And there was a question about the 15 seconds. How did you get to decide on 15 seconds?
Yeah. And so, you know what, I think So this is just something that was recommended when I started. I think as long as you're looking at something that has a linear slope, you're fine. If you want to use 30 seconds and it's linear over the whole 30, I don't think there's any reason not to do that. I think 15 was sort of a compromise before a place where the kinetics started to plateau. And so as long as you're picking a range where you're linear, as far as I know, in terms of calculating your rate there, you're fine.
Okay, and then Elena asked about freezing the commercial enzyme. Do you add any cryoprotectants or?
So I've done nothing. Seriously, all I did was I took the little vial they gave me. I added doubly distilled water in to get it to an activity that I wanted. And then I used some of it and there was just a ton left. And most of the time you have to dilute, even what I made, I had to dilute for the MDH specific activity assay. And so I just had so much left that I figure if I left it in the refrigerator for an extended period of time, it was going to die. And so I just shoved it in the freezer and I just put them in little Eppendorf tube aliquots. And so anytime I need a control, I just pull one out. It's still perfectly fine. Okay. That said, it's probably safer if you take it and put it in the same sort of cryoprotectant solution that we put the MDH we make in the lab in.
Okay.
Good. But again, I haven't, it's still kicking, so we'll see.
Well, if you're not using it that quantitatively, you just want to say, yes, right, my assay is working. It's less important. So,
yep. Okay, good to know. Thank you.
Yep.
Right.
Other questions?
Those were the questions in the chat. Anybody else have any questions?
Okay, if not, I'm going to stop sharing and let Amy share her screen and jump in.
Okay. Yeah, so I'll go to my share, but I was gonna just some really general things, some pointers about kind of how to talk to students about doing the calculations and about how we scale to larger things. I will say that we talked a lot this summer about the instability of oxaloacetate and how it's really best made up in water, which seems completely unintuitive to me, and using it fresh. And we have felt that our data have been more consistent this semester. So I think when you're working with students and there are all kinds of other variables, it's good to kind of, not play with the things that are that you can. We also changed our buffer to pH 8 from, it was a little bit more of a neutral buffer before. And again, that probably doesn't make a huge difference for MDH, but it may give you a little more margin when you're working with students who don't have a lot of experience. So I think we're sticking with that. All right, let me go ahead and share my screen.
Hey, can I jump in and ask a question real quick?
Yes, absolutely.
Yeah, Jones here. Regarding the quantification of the malate dehydrogenase, we kept ours at midazole after the elution, and when we ran a protein gel, we noticed that there were two distinct bands that were right on top of each other, one much fainter than the other. We assumed that our larger band, and it's about the same size, I think it's about 35 kilodaltons. It's a homodimer, I believe, right? So it's about 35 kilodaltons or so. So we assume the higher concentration or the higher intensity band, that's our malate dehydrogenase. We haven't used any immunostaining to confirm this. And in this case, I just wanted to ask, have any of you run protein gels to determine the purity of the purification?
So we've not seen two bands when we purified. There is an E. coli band. I think it's an outer membrane protein that runs right about that size. I don't know, anybody else had?
Yeah, so we run protein gels all the time, and I haven't seen two bands either.
Oh.
I will say there is a second band, a larger band around 70 or something that you sometimes see, especially with the mutants, that is probably some kind of chaperone that's probably upregulated in some of the mutants. And that does sometimes seem to come off with the I don't know whether it has a couple of histidines or because it's bound to the protein that's bound to the column that sometimes comes off with it. Has anybody else seen anything that's closer in size? I mean, that could be a small chaperone in too, but you said this is wild type, not mutants, right?
I think this was observed for wild type and a few of our mutants. So
Okay.
it seems like it's present in most, if not all of our strains that we tested. We tested 5 this past fall. Some of them, I think there was only one that was fairly clean, but you can still make out some very faint bands right above it. I asked Alice about this, and we couldn't quite figure out sort of what was causing that. I was wondering if it's also possible the concentrations that were growing these up at post-induction of the malodehydrogenases could have potentially triggered some unknown pathway that could have done some maybe post-translational modification that might have made it a little bit heavier. I don't
And
know.
these are and these are
just-
just BL21 E. coli, right?
Yeah.
One thing that we've noticed is if you put too much lysozyme or too much like DNase or something like that, and sometimes you can see those on the gels, like if students put too much in, or I had in my lab, they were using the wrong stock solution. So I don't know if that's possible, what you're mixing in with your lysis.
Okay. I mean, we do add lysozyme, we add protease inhibitor, and that's about it. Other than that, we just do the typical sonication and purification.
So if you're going to use your protein like immediately, like next week or in the next week, I don't add lysozyme or any of the protease inhibitors, and the proteins have been perfectly fine. So you could try that and just, as long as you're keeping them cold, you could try that and just see if that changes the banding on the gel. The other thing is when you pull your protein off the column, do you collect it in one fraction or do you collect multiple fractions?
We collected multiple fractions, and we keep them separate and then run it on protein gel to figure out which fractions then to pull together to run our assays based on purity.
And do all of those fractions have both bands?
Yeah, pretty much across all fractions.
The other thing that you could try to do is try to do a wash with a lower concentration of imidazole and maybe pull off the band that you don't want before you pull off your MDH. So you could play a little bit with the elution.
You might also double check your plasmid clone if you have like 2 populations in there, one that has some kind of deletion or something in it that's making a protein that's slightly different. I was getting double banding on the plasmid for a while, and I just got new plasmid from USD, and that went away. So I don't know.
Oh, okay. Yeah, because
those are unusual. It's unusual.
Yeah, these plasmids were provided to us directly from Ellis. I think Patrick, my colleague, picked it up.
I'm sure
2 years ago.
Yeah, I'm sure they're good. It's just that sometimes things, like 2 years ago, sometimes things happen in your freezer, in the fridge.
Yeah, we'll look in it. And thank you.
Yeah,
very much.
you're close enough. You could just drive down and get more, right?
Yep, very close.
Yeah.
All right, thank you.
Yeah.
Thank you.
Sorry. Anyone else? All right. I'm going to go back to this. So one of the things we've had a few questions about, and students are not always experts in Excel, even though you think they're so technically savvy. They don't always use spreadsheets a lot. And so various people in our community are using kind of different ways to kind of help them get going in Excel or use it to their advantage. And so you may want to give students something that's got a template, got some information already in it, or a step-by-step protocol that tells them exactly how to do things in Excel. So I just have kind of some suggestions here for things people have done, different levels of class. If you have an upper level biochemistry class, you may want the students to really kind of do a lot of the calculations and processing themselves. So one of the ways we do it is we kind of go over the calculation for a specific activity. So it's going to factor in, you need the attenuation or extinction coefficient for NADH, and you need the path length for your reading in the spec. But then we actually just put that into the template for them. So because we don't want to have, when I'm teaching Lisa mentioned this, Liz and I are teaching, we have 2 rooms of 24 students, so we have 48 students that we're working with at a time. And so you can't always catch everyone that made a mistake. So we're trying to give them some stuff. The other thing that we found that can be a real time sink is doing dilutions. And we have them do Michaelis-Menten curve, so they have to do a dilution series of their oxaloacetate. Even, I mean, it ends up being a really good learning activity for them, because the concept of dilutions is so simple, and yet actually figuring out how to calculate them can be the thing that takes the most amount of time in the lab. So often what we'll do is we'll do 1 or 2 with them, and then we'll, give them the pattern and let them do the rest. We also usually tell them to do the whole dilution series that you want. They're going to have a couple different dilutions of a stock solution. And so kind of explaining that this is going to be from your original stock solution, and this is going to be from a 1 in 10 stock solution, and this is going to be from 1 in 100. We put a lot of that into the Excel sheet, as well as try to explain it to them, just to kind of minimize the errors when they're actually doing it. I don't know if anyone's teaching intro level classes. I know Tam, who teaches intro bio, she just has a function where they plug in their change in absorbance and their protein concentration, and it gives them a specific activity. So because she just doesn't want to spend a lot of time with all the factors in that. So depending on what you want to do, I mean, you might just give them a template that just has the labels of their columns, and where they're subtracting or multiplying or dividing, and they can provide the rest. I don't know if anyone else has had any fun ideas with Excel and getting students going on Excel in their classes. Or any questions about that? And then the other thing I was going to talk about again is considerations for larger classes, or if you just don't have enough specs. And so we do not have 24 specs that our groups can be using. And so there are different ways you can work with it. If you, one thing you can do if you have a smaller class and not quite enough specs is just kind of stagger the timing, is have 2 different tasks that students have to perform, and half of them do the one that doesn't require the spec, and half of them do the one that requires a spec, and then you switch them. That's, of course, you have to be really conservative with how much time it takes them, because of course it always takes them longer than you think they'll take them. Our solution to that is to use a microplate reader. And of course, with the microplate reader, you can't actually follow them in real time unless you buy the $100,000 edition that lets you do all the robotics and follow them in real time. So we do a stop time assay, which means we run the reactions and then we stop them with sodium carbonate. And we run the reactions for about 1 minute or 2 minutes. That's about the amount of time the students can handle. And then but then they stop all the reactions, and we read all the reactions in one read. One of the things when you're doing stop time means that you have to have an NADH control. That's NADH buffer, but not enzymes. So it's basically telling you where your NADH starting was, because the only thing you're going to see is the endpoint reading of where your NADH ended up. This can be an interesting concept to get across to students. And a lot of times they'll do this and they'll do their Michaelis-Menten, and it'll be completely inverted, and they'll be like, I don't understand this. And I'll be like, gee, did you remember to subtract it from the control? And so that's things for consideration with the stop time assay. We do get pretty nice curves doing this, but it always feels like you miss some of the information by not seeing the actual the runtime. The other thing about when we do these in the microplate, we actually have them set up the reactions in Eppendorf tubes on their bench, because I worry a little bit about having them set them up in microplates, because they splash and they get into the next one, and you can really get a lot of confusing data. So, and they set them up in a volume of like 350 microliters. They then once they run that reaction and stop it, they put it into 2 different microwell plates. So we get basically a duplicate for pipetting errors. Those two should, wells should be really identical reads. And then if they really screw something up, they still have enough to do at least one more read in a microplate. So we've been doing that. They can kind of set them up and, as they get more comfortable with it, be running a few different reactions, at a time, just staggered 1 minute apart or something. And they get pretty good at that, and they can run, a couple of different Michaelis-Menten curves in a lab, doing it that way. So those are things that we do to kind of help work with large numbers. And any questions about any of that?
I actually had a question, and really for anybody out there, in terms of not a lot of equipment. We have one spec, and I will probably have 5 groups of students, 5 groups of 3. And I'm just curious how anyone out there has, worked with this or dealt with this problem. How much time should we devote to different groups who are running the spec to make sure that they have enough time if we're staggering the time? Any advice, I guess?
And we've had 2 specs for a long time, but if you only have 1 spec, even staggering will result in a lot of wait time for students.
That's my
even
concern.
Yeah, and generally it will cause a lot of frustration.
Yes.
We had 2. I mean, even with 2 specs with 4 groups, I think it was still kind of we were able to get by in Biochem 1.
Okay.
But I can't imagine only having 1 spec and trying to do it with 5 groups.
Yeah, and you can do the stop time assay in a regular spec. It doesn't have to be a microplate. Right. So you can have them all set everything up, run it, stop it,
That's
and
not a bad idea.
then they just go and read. And there's a little bit of a wait because some of them will be done earlier, some of them will be faster at pipetting, some of them will be slower, but there might be a little bit of a backup. But and then you also, kind of petition for more specs. we actually there are these little the little verniers that we use are not that expensive. Not we use, but USD uses. I don't actually have them.
So we do have some of those. I've never used them for anything like this, so I wasn't sure if they're capable.
Yep.
Oh, okay. I might have to look into that.
Yep. Yeah. And
Okay.
and I mean, Alice and Joe have protocols and everything. They're not on, we can send them to you if they're not
Okay.
on our
Yeah, I
site already.
I didn't find them on the website. So
Okay. Yeah.
Perfect.
Yeah.
That
absolutely.
would change things.
Yeah.
Thank you.
It's really nice for them to get to see it, if you have the resources for that.
Yes.
Yeah.
The other thing that you can do is, kind of depending upon how you want to spend your time with your students, is you can figure out what the concentration of enzyme is that they need in the cuvette.
Yeah.
So you can tell them exactly what to use, and then they're not spending, then in a half an hour they should be able to get 3 or 4 runs in and get some data that they can work with.
Okay.
And you can have sign-ups if you do something like that and have them sign up for time slots. Go ahead, Jones.
Oh, is it possible to kind of kill 2 birds with 1 stone and divide and conquer kind of concept? Have 2 of your groups work on, or 2 or 3 groups work on kinetics, and then 2 other groups, half the class work on kinetics, half the class work on some other characterization.
Protein gel or something.
Yeah.
And then
That's not bad.
split them, and then have them repeat and then swap so they get the same experience. And then that way you increase your N value for your data processing.
Yeah, I mean, you could do something like, one day half the teams do gels and half the teams do kinetics, and the other day it flips, 2 weeks apart, something.
That's a good idea.
Yeah, you got to be creative.
Yes, that's for sure. Thank you for the ideas.
I do have a question regarding the data processing. Do you do for the Excel? Do you use the linearization models for the KM and Vmax determination, or primarily, or do you use some nonlinear regression?
We have them estimate off of the Michaelis-Menten curve. We just, our experience, again, doing it at scale is that they just don't have enough data points to get good Lineweaver-Burke. I had one team that got really awesome Lineweaver-Burke. Their data were so good they went and did it because I didn't even tell them to do it. But most of them, it's just really not very good, and then it ends up being very different. So yeah, we just ask, we feel like you get a more reliable if you're kind of looking at the curve. But it, you know, if they have really good data, or if they have the time that they can just get a lot of data points, that'll make that more reliable.
And I did have a question regarding dilution of the enzyme. When you do the dilution of the enzyme to try to get that linear range of the rate, do you dilute it in water or phosphate buffer?
That's probably at least a question.
I usually dilute it just in the same phosphate buffer that I'm running the assay in.
Okay, all right. Is it so? Would it be so then in this case 100 millimolar, or would you try to dilute it down to about 50 millimolar so that when you add your enzyme, it's not going to affect the overall concentration of the phosphate buffer at the end?
You're adding such a small volume of that enzyme solution in the cuvette. It's only 5 microliters. So I just leave the phosphate buffer the way that it is.
Okay, that's fair. Yeah, because we ran into some issues with before. I don't think we saw on our protocol that we were supposed to dilute it in buffer. So we just ended up diluting in water, and we observed rapid decay of our rates over the span of maybe half an hour to an hour. Which is about the time it takes for us to go through all of the collect maybe 6 or 7 measurements, and prep the samples for the absorbance measurements. It wasn't until we talked to Alice that we realized that the buffer mattered a lot, and after we tried it, it became a lot more stable.
Okay. Yeah, and I would also make sure that you're keeping your enzyme solution in ice as you're sitting at the spec doing the assays. I try to keep, you know, once I've lysed my cells open, I try to keep everything as cold as possible just to extend the lifetime of the enzyme.
Right. Okay, cool. Thank you very much.
Yep.
Yeah, and there were a couple of things posted in the chat. Delphine said she's using Prism instead of Excel, and I keep thinking about that. I haven't ever converted to it. And then Chris had a couple links that that you might want to grab before we stop, that I'm looking up about fitting enzyme kinetics data and protocols. So take a look at those if you're doing Michaelis-Menten. Anything else? Other questions? Just about the end of the hour.
Any good suggestions for those of
Yeah,
you?
we got a lot of
that
experts in
assays?
here that haven't spoken up, so. Other thoughts? Well, this is great. And, you know, I think a lot of times just having a chance to kind of say, oh yeah, and I had that question and I didn't think of it. That's what we're here for. That's what Slack is here for. If you're not on Slack, let me know. I can help. I can give you the link for Slack. Yeah, I mean, anything we can do to help anybody's, and it's a great community, like I said, just because Lisa and I don't have all the answers, but between our community, we have a lot of answers. So please ask your questions. Anything else?
And will this be posted, Amy and Lisa, the recording?
Yes, we're going to
Okay.
post the recording on the website, on the MCC website. We can also put it in. We can also put it in Slack,
Okay.
and we'll post our slides as well.
Yeah,
Okay,
I think I'm actually gonna